Hello class, hope you're doing well today. This is the final in our series on the nature of science. So we're going to talk about graphing, data analysis, and just touch on the basics of what is biology. So let's get started. Hopefully you've done graphing before. I'm guessing that you probably have. But we do want to take a look at the basics of setting up a graph. So certainly we want to have a title. Your title should be a complete sentence that is fully descriptive of the graph, of the information that's being presented. You should always label the x and the y axes in terms of what they represent. The x-axis is where you'll put the independent variable. So the factor that we are altering goes on the x-axis. So that is what we are changing. On the y-axis, this is where the dependent variable goes, and that is what we are measuring in our trial. Finally, the scale of the graph should bracket your data. So as you look at our scale here, it goes from 0 to 600. That's within the range of our data. We would not want that scale to go, say, 0 to 2,000. Okay, there's no need to go that high. We simply want to bracket the data that we are presenting to give it the best representation. So these are all things that you should have whenever you do a graph. Now, in terms of the main types of graphs that we'll use, a bar graph will commonly be used. The bar graph is used if we're comparing two groups. So for example, as we talked about last class period with an experiment comparing two different types of fertilizer in growing tomato plants, a bar graph would be very good in that instance. Or in this case, here we're taking a look at how easy it is to fall asleep in different settings, whether you're in class, at work, or in bed actually trying to fall asleep. We're doing comparisons. And a bar graph can be very good for that purpose. The other type of graph we'll primarily talk about will be the line graph. A line graph is where we're trying to establish a correlation, typically. So here we're trying to establish the correlation between the complexity of passwords over time. So as time goes on, how is the complexity gone? And back in 1990, you could get away with something simple. And nowadays, not only do you need to include uppercase, lowercase, typically numbers, and also symbols into your password. So we're seeing a correlation here by using a line graph. So here's a nice example of a graph. Here we've got a very descriptive title telling us what it is, Indiana grain yields since 1930. We've got the yield, and also that is listed in units. Always list units. Here's our year, going from 1920 to 2010 in this case. Here are our data points, as we see, over that time period. So this is a very good graph establishing a correlation between these two factors. So a nice example of a line graph. And you'll see the blue line represents what's called a best fit line or a trend line. And so that's actually going to show us that linear relationship amongst the data points. Now, when we talk about interpretation, not just by looking at it on a graph, we typically carry out a process called statistics. And statistics is the science used to estimate the reliability of our data. So just by looking at our data, we can't necessarily infer as to whether that's good data or not reliable data. So we carry out statistics. And you might wonder, why do you need to know about statistics? It's because you are going to be consuming information. And if you're trying to follow information that is presented in a scientific light, it is presented with accompanying statistics. And in fact, much information that you receive even on a daily basis. In fact, you will be inundated with statistics over the course of this semester, simply because it's an election year. So poll data, for example, is a statistic. So ultimately, we want to be able to evaluate, is there a difference between two groups that we may be comparing? So we have to have an understanding of basic statistics. So let's take a look at an example. Here we have two groups of individuals. One receives a placebo lozenge, and the other receives zinc lozenges when they first start getting a cold. And we're going to then measure the number of days until cold symptoms are gone. And we see here that those with the zinc lozenges had 3 days shorter period of symptoms. So does that mean that zinc lozenges shorten the duration of symptoms? And that's something we want to be able to evaluate. And to evaluate that, we have to understand statistics. So let's take a look at some of these common statistics. The first common statistic that we look at is the mean, often referred to as the average. You'd usually know this as the average. This is where we just add up the data points and divide by the number of data points. But what I want you to realize is that the mean doesn't tell the whole story. So for example, here we've got a data set of individuals. And if we determine what the mean is for this, we add those up and we get 25. We divide by 5 because we have 5 data points for an average of 5. Now let's compare that to another group here. What is the average of treatment 2? You'll notice that the average is also 5. So does that mean that these two groups are the same? And if you think about this, the means are the same, but if you look at those data sets are very different from one another. And that treatment 1 varies from 1 to 9. There's a large degree of variation. Treatment 2, there is a small degree of variation. And knowing how much variation is present can be very insightful into understanding and properly interpreting the data. So to present an understanding of how much variation is in the data set, we use what's called a standard deviation. Standard deviation is simply a measure of variation in the data set. So it's going to tell us, like here we're taking a look at graphs of the average height of individuals, whether they are male or female. And we'll see that females typically have less variation in height than males. Therefore, males would have a greater standard deviation in terms of their height. Females would have a lower degree of standard deviation in height. So it really tells us the width of a curve if we're presenting our graph as a bell curve. So if we take the mean, or average, and we add one standard deviation, and we subtract one standard deviation, that would include 2/3 of the total population. If we go farther than that and we add and subtract 2 standard deviations, that will actually account for 95% of the population. So this is going to give us some insight into how standard deviation is sometimes used. Another common statistic that is used is the standard error. Standard error is very much like standard deviation. However, it takes into account the sample size. So, for example, Let's say we wanted to estimate the average height of students at Jackson College. And I randomly chose 4 students out of this class to get an estimate of the height of students at Jackson College. So would that give me an estimate of the height of students? Yes, it would. Probably within a foot, it would be correct. Okay. However, I think you would agree that if I chose the whole class and took the mean height of all the students in the class, that would give me a better estimate of the height of students. And so that's where sampling or standard error comes into play. It takes into account how many individuals or how large of a sample size you have. And when we use the term error in this case, it doesn't mean a mistake. In this case, standard error is referring to chance events in sampling, that you drew a odd sample. So for example, if I chose my small sample size of 4 students, maybe I chose 3 females and 1 male. Therefore, my sample was biased. Not intentionally, but just because it was an odd sample, not reflective of the percentage of males and females at Jackson College. So if you have a larger sample size, you're less likely to have these unusual sampling effects, and thus a larger sample is more reliable. So what we do here, and I'm not going to have you calculating this by hand, but just as to be aware of this, you take the standard deviation and we divide it by the square root of the number in the sample. So as sample size goes up, the standard error will decrease. Many graphs will actually show the standard error by using what are called standard error bars. One other statistic that is often used as a step off beyond the standard error is a 95% confidence interval. These are very commonly presented as a statistic. So this will present the range of data that we are 95% certain that the true value exists. So if we if we came up with a height, mean height of students in our class of, say, 5'8", for example, or 5'7". We'll say 5'7". The 95% confidence interval would be a range of data that we're 95% certain that the true average height of students at JC would exist. So typically it's presented again as plus minus the mean plus or minus some amount. So we actually determine this by taking the mean plus or minus 2 times the standard error is how we would actually calculate this. Now, where's an example of this? Well, here is an example of a poll that was conducted some years ago. And this was a poll of Americans, and they were asked a true or false question. True or false, the continents have been moving over millions of years and will continue to move. And as they did this poll, they came out with a mean that 77% said true, and the remainder said false. And in this study, there was a 95% confidence interval of plus or minus 3%. So what this is telling us is at the low end, we can be 95% certain that the low end would be at 74%. And at the high end, okay, we got that 74 by taking 77 minus 3%. Then we take 77 plus 3%. So we would be 95% certain that the true percentage of Americans that knew the correct answer to this question was somewhere between 74 and 80%. And again, these are very commonly used statistics that are presented to the public. So when we're looking at data, let's go back to looking at our control group versus the group that received the throat lozenge, the zinc lozenge. Here, let's say we had a very large sample size, and thus we had a small confidence interval. All right. In this case, we notice that we're 95% certain that those individuals that would take a zinc lozenge would be over their cold between 3 to 5 days. Those that did not take a zinc lozenge would be over their cold in somewhere between 7.2 and 9.2 days. All right. So in this case, since these ranges do not overlap, we would say that there is a difference. However, let's say we had a small sample size, or maybe we just had a large variability in how long it took people to get over their colds. And we had a large amount of variation in our data sets. Now you're going to notice that these data sets potentially could overlap. So it could be that those people not taking a cold, their true mean could be down near 6 days. And for those that were taking zinc lozenges, the true mean could be up at 6 days. Since these error bars overlap, then we could not say that there is a difference between those two groups. So let me show you an example how this can be used that you'll be seeing. Whoops, we've seen that slide already. As we move up towards November, we're going to be seeing a tremendous amount of poll data. And polling data is presented as 95% confident with 95% confidence intervals. So let's say we did a poll of support in the upcoming presidential election. Candidate A was at 48%. Candidate B was at 45%. And based on the confidence interval is based off of the number of individuals that were sampled in that poll. So as the sample size goes up, the confidence interval decreases. So in this was actually in this poll, we had 426 likely Democratic voters. This was actually in from primary data at one point, and the confidence interval was plus or minus 5%. So in this case, if we're to properly interpret this data, what we would say is that we can be 95% certain that the true support for candidate A is somewhere between 48 minus 5 and 48 plus 5%, so somewhere between 43 and 53%. Candidate B is somewhere between 40 and 50%. So we take 45 minus 5%, 45% plus 5, and so we get this range of data here. And keep in mind that when we look at national polls, this is a representative of what is not uncommon for national polls. So here, many national polls may be conducted from a survey of typically 1,000 people or less. Now, as we interpret this data, what we see is that these data ranges overlap. There's a lot of overlap between these, and since the data sets overlap, we could not say that there is a difference. On the news, they should present this, that there is a statistical dead heat, is the term that they would use from this poll data. Now let's consider, let's say they did a much larger sample size. So if we did a much larger sample size, in this case, let's say we got the same data, but there was a tremendously larger sample size, and the 95% confidence interval was plus or minus 1%. Now in this case, as we looked at this, we would conclude that we could be 95% certain that the support for candidate A was somewhere between 47 and 49%. The true support for candidate B would be 45 minus 1%, 45 plus 1 is somewhere between 44 and 46%. Now we'll notice that these data ranges do not overlap. So there is no overlap between those, and thus we could conclude that there was a significant difference in the support of one candidate versus the other. And that difference is primarily due to sample size. So that is how we make use of 95% confidence intervals. Now, one other statistic that I do want to introduce, and this one you're going to have to kind of just brute memorize, and this is something called a p-value. If you go to read scientific findings, or a science journal or a science article, and you're looking at, say, a graph or a table, and you want to identify if there's a significant difference between groups, you often need to examine what are called p-values. And p refers to probability, and it is the probability that there is no difference between the two groups. And I realize this can be kind of confusing. This gets back to some of the basics of how we actually do science. We don't need to worry about that. So, and these p-values will range between 0 and 1. So if the p-value is 1, if we present that as a percentage, that's 100%, that means there is absolutely no chance that there is a difference between the groups. So if it's a high p-value, that means there's little chance that there's a difference. If the p-value is very low, as it approaches 0, when we reach 0, then we are absolutely certain that there is a difference between the two groups. Now in science, we've kind of set a agreed upon limit where we truly accept that there is a difference. And that's the 95% certainty level. So if the p-value is less than 0.05, we would say that yes, there is a difference between those two groups. So if the p-value came back as 0.01, we would say yes, there is a difference between those two groups. If the p-value was 0.95, we would say no, there is no two groups. And what this is saying at 0.05 is that we're 95% certain that there is a difference between those two groups, or essentially saying that there is a difference. So at 0.05, there's a 95% chance that there is a difference. So when we use p-values, you want to look for small values. And later on this semester, we'll look at scientific data that presents the data in terms of p-values. So we'll want to think about that. The last thing that I do want to touch on before we move on with this semester is that as we proceed from here on, we're really going to be looking at science in the realm of biology. So we really need to think about what is life, because biology is indeed the study of life. So as we consider life, how could you define what is life in a single sentence? Well, it's really a difficult thing to do. We can't really define life in a single sentence without saying, comparing something that's alive with not alive. So in science, in biology, what we do is we describe life by using a set of characteristics. So these are the characteristics that all living things share. So here we're looking at those characteristics shared by all living things. So one of those characteristics is that all living things are composed of cells. They either are a single cell or they are composed of cells. And that is one of the hallmarks shared by all living things. Living things reproduce themselves with the genetic material of DNA. So all living things that we've ever examined have DNA. Sometimes scientists get into some discussion as to whether viruses should be considered living because some viruses do not have DNA, such as coronavirus does not have a DNA genome, genetic material. But all living things, whether they are plants, fungi, animal, bacteria, use DNA as their genetic material. All living things obtain energy from their environment, either by eating other things or by using light or digesting dead or decaying materials. All living things sense and respond to their environment. So if you go outside and you see a little bunny sitting there and you clap your hands, it will respond to that sound typically by either becoming more alert, maybe it will try to run off. But realize even bacteria sense and respond to their environment. So if their environment changes, that will result in various genes being either turned on or turned off, and they will respond to their changing environment. All living things maintain a stable internal conditions. This is referred to as homeostasis. So, for example, you are maintaining stable internal conditions. So, for example, if you think about internal conditions that your body has to maintain, your body has to maintain a certain concentration of oxygen in your body. And if you don't maintain that homeostasis, what happens to you? Bad things happen, right? So we could think about the amount of water inside our bodies. If you lose too much water, you become dehydrated, and you can die from that. And you might be surprising, but if you actually have too much water in your body, that can actually dilute some of the salts that are in your body that are necessary, and that can actually lead to death of an individual. Body temperature is a homeostasis that we as mammals maintain. So there are a tremendous array of homeostases that we maintain. So you have to have certain salt concentrations. Sugar levels have to be maintained at certain levels. So various minerals, calcium, iron, and the likes have to be maintained at certain levels within our bodies. And all living things have to do this. Even bacteria have to do this. Plants have to do this. Living things can evolve. So as populations, living things evolve. So they change from one generation to the next. So as individuals cannot evolve because you cannot selectively change your DNA over your lifetime. However, populations can evolve. Those individuals that are not as well suited will tend to die off, and those that are well suited to their environment will have a greater chance of surviving and passing their genes on to the next generation. So those are some of the characteristics that are shared by all living things, and we certainly should be aware of these as we move forward. So that wraps up our last bit on the nature of science. Again, if you have questions, please don't hesitate to ask. And again, have a great day.